Next one is structure of ovule or megasporangium. So as we are discussing that megasporangium or the ovule which are present inside the ovary, and the number of ovules in one ovary is not a constant number in each and every plant. So it may be ranging from one ovule to numerous ovules in each ovary. Generally, if you take the examples of wheat, rice, mango like plants, so one ovule is present in each ovary, whereas numerous ovules are present in each ovary can be seen in plants like watermelon, papaya, like many such examples are there where one ovary contains many ovules. And where does these ovules are present inside the ovary? In the earlier lessons, that is in the flower lesson, you might have discussed that ovules are developing inside the ovary on a specialized cushion like parenchymatous tissue that is called as placenta, and the pattern of arrangement of the ovules inside the ovary is called as placentation. So here let us come to the structure of ovule, and ovule is otherwise called as integumented megasporangium. Presence of ovule is a characteristic feature present in both gymnosperms and also in the angiosperms. That is, it is a characteristic feature present in the flowering plants, that is phanerograms or spermatophyta group of plants. Each ovule contains a stalk like structure which help in attachment of the body of the ovule to the placenta. That stalk of the ovule is called as funicle or funiculus. Funicle or funiculus. And if you see the structure of this ovule, basically this is a globular part considered to be the body of the ovule. So this is the stalk, and this part is considered to be the placenta on which the ovule is developing, and this part is called as funicle or funiculus, considered to be the stalk of the ovule, and this is the body actually. The ovule body of the ovule is ovoid in shape, and the body of the ovule and the funiculus are attached at a point. That point of attachment of body with the funiculus is called as hilum. The point of attachment of funicle and body of the ovule is called as hilum. Sometimes the funicle may show overgrowth onto the body of the ovule and it form a ridge-like structure. That ridge-like structure which is formed by the funiculus is called as raphe. Raphe is a ridge-like structure formed because of overgrowth of the funiculus onto the body of the ovule. And raphe can be clearly visible in the anatrophous type of ovule. Then if you observe the body of the ovule, it is nearly oval in shape, and it is protected with layers of tissue. That protective sheets are called as integuments. So can we see here two integuments are shown in this diagram. So this one is a outer integument and this one is a inner integument. And one more thing, so the integument, the outer integument is completely enclosing the ovule, but at one region it is leaving a small gap. That small gap is called as a micropyle. The integument completely encloses the ovule, but a small gap is left out. That small gap which is left out at the anterior side is called as a micropyle. And of course, the inner integument is also leaving a small gap there. And opposite to the micropyle, the region where the integuments and the nucellus are fused, this part is called as a chalaza. Opposite to micropyle, the posterior side we call it as a chalaza, and in the ovule wherever the micropyle is present, that end is called as a micropylar end, and wherever the chalaza is present, we consider this side as a chalazal end. Generally chalazal end is called as a posterior side, whereas the micropylar end is considered as a anterior side. And inside this integuments we can see a diploid tissue. This diploid tissue is called as nucellus. The diploid tissue is called as nucellus, which is nutritive in its nature, and inside the nucellus we can also see the presence of the embryo sac. So this embryo sac is otherwise called as female gametophyte, and this embryo sac contains 7-celled or 8 nucleated structure. So this is called embryo sac. These are the parts that we can see in the structure of an ovule. And one more thing here it is not assured that all the ovules should contain 2 integuments. So there are some plants in which the ovules does not have the integument also. So based on presence and absence of the integument and the number of integument layers, so 3 types are present. So let's see the 3 types of ovules based on the integument numbers. So the first one is ategmic ovules. Ategmic ovules means here the nucellus is not covered with integument layers. This type of ovules are called ategmic ovules, which can be seen in Loranthus like examples, whereas the second one is unitegmic ovules. Unitegmic ovules means the ovule in which a single integument is present. So this is present in gamopetalae of dicots. So this type of character can be seen in datura like examples, and also in sunflower, triadax like examples. Whereas the third one is there, that is bitegmic ovules, where the nucellus is enveloped by 2 layers of integuments that we have seen in this diagram. So this condition is called as bitegmic, where the outer integument is called outer integument layer is present and the inner integument layer is present. So this type of character can be seen in the monocots and polypetalae of dicots. Polypetalae of dicots. So these are the 3 types based on the number of integument layers present in the ovule. Those are ategmic, unitegmic and bitegmic. So one more aspect, character related to ategmic is, so as we said Loranthus is present. This type of character can be seen in the parasitic plants like in Loranthus, and some other examples we can consider here. So the most common type is a unitegmic condition, where single integument is present in most of the dicots, and of course we can also see the bitegmic in all the monocots and the polypetalae of dicotyledonous group of plants.